We've got 15 minutes for questions, and as you understand, the program today is such that this morning introduction and a very regulatory focused, and later sessions are more technical, if I put it that way. So perhaps if we could focus the discussion on the regulatory aspects, need for regulatory guidance, in light of what you've heard from all speakers, and maybe just to stimulate the discussions, and it's a question to the panel, but also to you. In your experience, and I'll start with you, Rob. In your experience as chair of scientific advice, you deal with 30, 40, 50 advice each month. What's the most common practical problem that you encounter from industry and that you have to address that you think could be solved or helped by further clarification in guidelines? You said yourself that you're not even sure this morning when you looked at slides again that if there needs to be an update in Europe, but if leave that apart, what do you think would be more helpful? And then I think I will ask you the same if there's something that you think is clearly missing that you find helpful from industry side. And then I'll ask the other panelists before going to the group. Yes, Rob, do you have any views? It's a tricky question.
Yes, of course.
Yes, that's why I ask you.
First reflection is how much synergy, how much agreement there was between the different positions expressed across a number of topics. I think I only heard one potential point of divergence, and that is actually the answer to your question. But I'll give two answers. So the first area on which more thought would be particularly worthwhile, I think, relates to this word claim. What does it mean? Does it mean the same thing in different regions? I think from Kathleen's excellent talk, the only discrepancy, divergence that I saw with the European view is on the extent to which we're trying to cover the secondary endpoints in the not in the family for alpha, let's say, and this is clearly an issue when giving scientific advice to companies. It's rather clear when a company has already had a discussion with FDA or is predicting what FDA are likely to say, and they construct the endpoints in a different way, and the alpha or the considerations for multiple testing across the endpoints is considerably more extensive. And that's not to say one's right or wrong, because I think Norbert explained very well that there may potentially be a different environment. But I think that's one point that perhaps needs some further reflection, and other speakers and Norbert highlighted that nicely. The other point that comes across, both from the talks and from work at scientific advice, is whether we have the proper communication between the different disciplines that make a contribution to this. And I'm absolutely sure that in the sponsor pharmaceutical companies, as well as in the regulatory agencies, there is dialogue between the statistician and the medic. And we do this for most of clinical trial planning. We do it for inference. We do it for decision making. But I'm not actually sure we do it as well as we can for control of multiplicity. And again, these ideas of the pharmacology, how many pivotal trials have we got? And when you put the totality of evidence together, what's the strength of our evidence from a statistical perspective? And I absolutely agree, and you explained it really very nicely, much better than I did, that avoiding arbitrary hierarchies. And I don't like arbitrary hierarchies. And we're guilty of this on the regulatory side. You must have a hierarchy for your secondary endpoints. Well, why? If they're all exploratory, if they're all supporting the one label claim, a hierarchy doesn't make any sense clinically or methodologically. So I think that there's some discussion around what's a claim. There's some discussion around communication and making sure that what's sensible from a pharmacological and a drug development point of view is actually what happens in the statistical considerations.
Yeah, I think one of the probably the challenges is, I mean, definitely my talk was kind of on a nice high level idealistic thing, and it's always difficult when you bring a specific product to a specific team. How can we agree? And I know that I think at the agency, I know usually the statisticians and the clinicians within a team, I think, tend to work very closely together, but I suspect you often get, if you work across therapeutic areas, I suspect you often do come across slightly different viewpoints depending on which teams you end up working with. And I think at the agency, we're trying to work on that, but and that's always a challenge. And I do think that one of the points that Norbert brought up was that probably we do have a lot of concerns about how marketing claims go out, and that I think often influences, we're kind of wary of letting a lot of things get into the label, though actually advertising is not restricted to what we put in the labeling. It's basically, if it's done in a well-controlled clinical trial, usually that means it's available for labeling, but that often, or for marketing. So that's often challenging, that we have to kind of think of how it's going to be used, and it's really hard on the individual cases, because particularly clinicians would love to see more and more endpoints. They're always more convinced, yeah, give me 3, give me 4 endpoints. If you win on 4 endpoints, I'll be really convinced. But of course that just, then the downside is, of course, they're often at the end convinced on 1 or 2 endpoints, but we've set up the hypothesis framework that the study only wins if it wins on all 4 endpoints, and then we're kind of stuck trying to salvage the study where then the clinicians, oh yeah, this looks great, but we've messed up our multiplicity plan. So we need to come to more consensus and really get to the point of what we need to know so that we don't have to backtrack and try and salvage our studies, because it comes from both ways.
This is not on, sorry. It's okay. Any reflections from your side? No? Norbert, anything?
Yeah.
There's no pressure, but if there's any reflection. Okay, so if I...
Okay. Yeah, I mean, well, Rob already mentioned the issues we get in scientific advice. I mean, what I, may add here. Well, as you said, well, we often get these questions as, would you accept the following procedure? Primary, secondary, adjustment like this, etc., etc. And then sometimes, well, the discussion is a bit controversial. Some say, okay, yeah, it's nice, but I'm not sure whether you really need it for some secondary endpoints. And then the problem that may arise is in an assessment. Imagine that multiplicity procedure was applied in secondary endpoints, and then you don't get a significant result for some of these secondary endpoints. But well, it seems convincing, but you don't get a significant result as planned. And then how do you deal with this in the assessment? Because well, it failed according to the procedure, but if it was not well required, and well, some people think it was not required, and other people think it was required, then there might be a controversy in the assessment whether this is failed or not failed. So probably this is one, let's say, problem we still have and of course, the question on whether we accept multiple SAPs is asked several times, and I think we will probably come to a unified answer here.
Okay, thanks. From your side, yes, please. What do you think is lacking, if I put it that way?
I was wanting to explore a little bit more this idea about multiple SAPs in multiple regions. You know, usually like the clinical trial is powered for the whole world. You might have 80 countries involved in your study. It's not powered for each region. So we typically adjust the regions just by maybe looking at them as exploratory, but, you know, I guess a little bit more understanding of what we're supposed to do in that regard, because we're not necessarily planning to have power in each region.
Yeah.
And so it's not addressed in terms of multiplicity then.
No.
But
Rob?
there's two aspects, as actually a number of the speakers described, and it's on this slide that's on the screen still. What you're talking about is the bottom one, consistency between regions, or what's the effect in the European population. And nobody is trying to say you have to have a statistically significant effect in the European population. There's guidance documents, ICHE 5 and a CHMP guidance document about extrapolation between different regions and when does it make more sense, when does it make less sense. And that's to try and help guide how much evidence should you have from a European point of view and how well can we extrapolate from other regions. But the vast majority of global trials, we just look at the primary contrast, yeah? And unless there's a reason to think that doesn't apply to Europe, that's what you use. Now, what we're talking about in terms of multiple SAPs is where, for example, the FDA want one primary endpoint and Europe want a different primary endpoint, and maybe Japan want the third primary endpoint, yeah? And then we can get you to run 3 trials, one for each region, but that seems a huge waste of resource. So what are the other possible solutions? And having one trial, but with different analysis plans, so a different primary endpoint in one plan, allows you to address the requirements of all 3 regions without having to run multiple studies. So that's the situation we're talking about. And Dr. Heder asked a very good question as to how then to publish the results in that situation, and that certainly is one of the complications. But I would hate to think we got near the situation of multiple protocols for a trial. I think a trial should have a protocol, and the protocol should say the primary endpoint, and the primary endpoint can be used for publication. The fact that you then have different analysis plans to address regulatory requirements, I think, is a slightly different issue.
Yes, please.
So in terms of what's missing, I think I'd echo the comments that Rob's made already in terms of this. I'm a statistician. This feels like an area, there's a community, we've done lots of work ourselves, and we're imposing it on the physicians we work with. And within the sponsors, I can tell you physicians don't understand why we're doing it and why it has to be so complicated. And my experience is that it depends how far we go with this. When I'll return to this later, because I think we're in danger of going too far. But if we go down a line of the way things seem to be going, I think there's a danger. And the reaction from many physicians I work with is, this is, to be frank, their reaction is, this is some statistical mumbo jumbo. That's what the statisticians do. And I can ignore it because when I get the data, I'll just make some common sense clinical decisions on them. So in terms of what I think's missing, I think is the engagement from the statistical community more broadly, as if we're going to go to all this effort, that actually we get buy-in with the people who are going to are part of the decision making ultimately.
Can I build on that one? I think that's a good comment. As a non-statistician, I hope you excuse me, Rob, but I think it's a good comment. Can I build on that? Because I think that's the two aspects of that. That's, to me, is a particular concern also when we start dealing with safety problems. And then it's not only clinicians, it's the public and patients. And on this slide, it's the multiplicity issue and safety assessment, which is, to me, is a decision making sometimes more difficult than the other ones, because I can go to Rob and others and ask in efficacy, but in safety, it's to me it's much harder. You know, you can't just say, well, you know, we test. Patrick and I were doing a registry study where we're testing 30 endpoints. One of them will of course at least fall out there, but we're going to say, nah, but that we don't care about that adverse event. It wasn't caused by the drug because we were adjusting our p-values now. It's not be credible. People that have reported adverse events, because that's why we're doing the study, they're going to say, well, no, there you go. They're just trying to hide the true bad drug here. Typical regulators defending industry, hiding the true adverse events behind the statistical mumbo jumbo. The other thing, perhaps related to this, is I understand when it comes to claims, that we don't want to be, that we want to be cautious and we don't want to allow marketing of incorrect claims. But to a decision maker, i.e. patients or prescriber, it may be of value to have a broader picture. And how do you combine that? How do you inform them of the totality of the data that we have, the totality of the data that we ourselves as regulators think are so important when we take the benefit risk discussion, if we're too restrict when it comes to claims? Fortunately, we run out of time at this moment. But I want one more question.
Let me make a comment, which is, I know from my long experience, more and more the tendency comes up that every medical doctor doesn't expect that somebody understands all the details, pharmacological and all these theoretical details. But it seems very, very clear that any statistical method which is not understood in 3 minutes is rubbish. And I think this is a really challenging thing, you know. It's very selfish from one area to the other. I was always handling problems in discussion with medical doctors, but the self-consciousness that we only can propose things which can be understood by any layman in 1 minute is absolute unacceptable.
Last comments from behind. Quick one, please, Paolo.
Yes, on the top, Paolo Tomasi from Pediatric Medicines here. On the topic of two different endpoints for the same trial, I have seen this situation in a few occasions in pediatric trials with the FDA. But in all the cases, the two measures were likely to be highly correlated, in fact. Now, my limited understanding of statistics is that multiplicity is more of a problem when the measures are not correlated, because if they're 100% correlated, there is no multiplicity problem at all. Now, my question is, how do we quantify this? How do we quantify the reduction in the risk of type 1 error if the two measures being studied are highly correlated between themselves?
Is that something comes back at more technical session? Yeah, well, I've been told that they will come back to that in a more technical session. So unless you want a more regulatory aspect from FDA specifically, was that the question? Then I can ask Kathleen if you have any immediate reactions.
And I think that kind of goes into a little bit of why we generally are fairly comfortable with doing agency-specific plans. Is it's not likely to really cause much of a problem in terms of actually interpreting the trial, because they do tend to be fairly correlated.
Rob?
But equally, if in this situation, if the company just pick one of the endpoints, so you have maybe the FDA as primary and the European as a key secondary, whatever that means, if they're so highly correlated, the likelihood is that you win on both anyway, and you avoid the complication. And that's what, there's different strategies for approaching the problem, I guess it's clear. You are, just quickly on your safety comment, just to say there is a reference in ICHE 9 to the multiplicity problems associated with safety assessment, which of course deals with both the type 1 error that we're mostly talking about today, but makes a special reference to type 2 error, which is even more important from the regulatory decision point of view, of course. And I guess the final comment, just for people to reflect on, when we're talking about labeling and your comments as to what's relevant to the prescriber and to the patient, which is the wording in our SMPC guideline and how we tend to think about it, a p-value of 0.06 is not uninformative. We have a dichotomy to make our lives easier for decision making, but 0.06 is not uninformative.
That was my non-statistical point, yes. I agree with that.